Welcome back, everyone, and welcome our next speaker. Before we move to the presentation, we would like to kindly remind you, if you could please, again, share your questions with us so we can discuss them at the end of the day at the panel session. And right now, welcome our next speaker, Dr. Eric Rawson from Messiah University. The floor is yours.
Well, hello. I hope you can all see my slides and hear me well. I want to congratulate the conference organizers for this fantastic conference and especially thank the technological people working behind the scenes to make sure that this all works so very, very well and smoothly. My task today is to talk to you about the effects of creatine supplementation on brain health. I think the best place to start any lecture on creatine and creatine supplementation is with a brief history lesson. So creatine was actually discovered in the early 1800s, with the first paper being published by Michel Eugene Chevreul back in 1832. Creatine was very, very heavily studied for the next approximately 100 years. And in fact, there were so many publications on creatine, the nutrient, in the 1800s and early 1900s that there's actually an entire textbook published on creatine and creatinine metabolism with over 500 references. So creatine research is certainly not new. Creatine supplementation is not new either. The great chemist, Justus von Liebig, actually marketed and sold an extract of meat supplement back in 1847. This was obviously a concentrated creatine supplement. So creatine supplementation is not new either. Additionally, research on creatine supplementation isn't that new either. The first publication appears in the literature on human supplementation in 1926. Alfred Shanoughton and Lauren Guy published this paper where these two individuals ingested creatine supplements and with urinary creatine measurements were actually able to demonstrate that when humans ingest creatine, not all of it is excreted. Some of it is retained in the body somewhere, although this study predates the muscle biopsy technique by several decades. So we've been studying creatine for a long time, and we've been studying creatine supplementation for a long time too. I think probably the two most important studies in the world of creatine supplementation research are these two studies, and one of them has already been mentioned several times. The groundbreaking research in the modern creatine supplementation era begins with the study from Roger Harris, Karen Soderlund, and Eric Hultman. And that's the 1992 paper published in Clinical Science, shown here on the left. But the paper from Deschent and colleagues in 1998 was equally as groundbreaking because this really marks the beginning of research on creatine supplementation and brain metabolism. So what did we learn from these two papers? These two important papers helped us build the foundation for all of the work we did today. From Roger Harris's 92 paper, we learned that oral creatine monohydrate supplementation does, in fact, increase muscle creatine. And we learned that although there is a great amount of variability, on average, one could expect about a 20% increase in skeletal muscle creatine. We learned from this first investigation that those of us who have very low muscle creatine levels have the largest response to the supplement, Or in other words, if you have very little creatine in your muscles, when you take the supplement, you have the largest increase. Also, Roger's paper taught us that exercise actually enhances muscle creatine uptake. The paper from Deficiencies and colleagues. This was the first paper to demonstrate to me that when humans ingest creatine monohydrate, they do in fact increase brain creatine levels. We learned from this paper that this increase is about, on average, 10%. And today we might argue that that's a little bit larger than the increase actually is. But from this paper, we learned that the increase was about 10%, or at the very least was certainly less than what happens with skeletal muscle. And we also learned another important point. There are regional differences in brain creatine uptake. When it comes to skeletal muscle and the muscle biopsy technique, we tend to assume that that tiny bit of muscle that we remove and analyze represents the entire muscle. And I think sometimes that's correct. But with the brain, and especially with creatine metabolism, things are very different. There are regional differences in basal levels of brain creatine, and therefore there are regional differences in creatine uptake following supplementation. So these two papers were important starting points for all of the work that we do today. Now, creatine supplementation research has evolved from predominantly sports science research to predominantly patient population and older adult research. Recently, researchers have been focusing a great deal on the effects of creatine supplementation and brain health. If you are very interested in this topic, there are a number of excellent reviews in this area, and this goes beyond the ones you see on the screen. As part of this conference and as part of the Nutrients special dedicated issue to creatine supplementation, we have a review called Creatine Supplementation and Brain Health. We've published an updated version in 2022, and there are other excellent reviews as well. Our three narrative reviews for your interest are, they're all narrative reviews. There's also an excellent systematic review published in 2018 in Experimental Gerontology by Averginos and colleagues. Now, I need to begin the content of this talk by reminding everyone that muscle and brain are not the same. And these differences are revealed over and over again during this presentation and during a thorough analysis of this body of literature. For instance, 95% of the creatine in our bodies is in our skeletal muscle. About 5% or less of the remaining creatine is stored in our brain. Creatine in skeletal muscle and brain is really used primarily for fuel and as an energy source to replenish ATP. One important difference between muscle and brain is that skeletal muscle does not manufacture or does not synthesize creatine. Creatine is synthesized primarily in the liver, the kidneys, and the pancreas, which means that skeletal muscle is essentially designed to take up external creatine or take up exogenous creatine. Brain is different. The blood-brain barrier is there for a reason, and the brain is very protective against external or exogenous substances, including nutrients. The brain has the metabolic machinery to synthesize creatine, and potentially exogenous sources of creatine, like dietary creatine or supplemental creatine, might be more difficult to get into the brain. The brain may be a bit more resistant to exogenous creatine, while skeletal muscle was really designed to take up exogenous creatine. We know that skeletal muscle responds easily to changes in creatine levels in the diet. For instance, if I begin a vegetarian or a creatine-free diet, But the small amount of information available suggests that brain creatine levels may not respond the same way to a reduction in dietary creatine levels. More on that in a few moments. I do not believe that skeletal muscle creatine changes in response to exercise training. I know a lot of textbooks disagree with my point, but I have not seen any conclusive research saying that high-intensity exercise training increases muscle creatine levels. So I don't think muscle creatine responds to exercise training. I don't know if brain creatine levels respond to either exercise training or, for lack of a better expression, brain exercise training. There is some literature to indicate that increased brain activity may actually alter brain creatine levels, but we really don't know much in this area, and we certainly don't know how supplementation would fit into the picture of increased or decreased brain activity and brain creatine levels. From a measurement perspective, from a researcher's perspective, when muscles are at rest, we generally believe that muscle creatine is stable day to day. So if I were to measure your muscle creatine today or tomorrow or in the morning or in the evening, as long as you've been resting for a few minutes, it's generally believed that skeletal muscle creatine is constant. I do not know if this is exactly the same for the brain. And one of the reasons is that it's very difficult to measure brain creatine levels in living human beings. There just aren't that many laboratories that have the ability to take this measurement compared to the numerous laboratories around the world who do muscle biopsies and can accurately measure muscle creatine content. So there are a tremendous number of differences in brain creatine and muscle creatine in terms of metabolism and in terms of measurement and in terms of what we really know versus what we really don't know. And here's a good example. When it comes to supplementation of creatine, we know a lot about the skeletal muscle response to creatine. Now, notice on my slide, I'll take a moment to go on a small tangent here. I have monohydrate capitalized, creatine monohydrate supplementation. About 99% of the literature on the safety and the efficacy of creatine supplementation really focuses on creatine monohydrate supplementation. So when I say creatine supplementation, I really mean creatine monohydrate supplementation. In terms of skeletal muscle, if you were to ingest about 20 grams of creatine per day for about 5 days, or perhaps 3 to 5 grams per day for about 30 days, we would expect approximately a 20% increase in muscle creatine levels. Remembering that there are some low responders, some moderate responders, and some high responders. In terms of brain creatine response to supplementation, 20 grams per day for 5 days may increase muscle creatine levels. And this increase may be about 10%. Why do I say maybe? Because there are far fewer studies on the brain's response to supplementation than there are creatine supplementation studies that have to do with skeletal muscle. So I have a number of studies that used about 20 grams per day for 5 days, and I have a few studies where the increase was about 5 or 10%. But one of the weaknesses of this body of literature is that we really haven't refined the exact supplementation protocol, not in terms of dose and not in terms of duration. For now, most of us are using what we know about muscle creatine in terms of dose and duration and applying that to the brain. But there's a lot more work to be done in this area. So what happens if you do ingest creatine and we measure your brain creatine or your brain phosphocreatine? If you have a real interest in this area, again, I'll refer you to some of the more recent reviews that have focused on the effects of creatine supplementation on brain creatine and cognitive processing. There are 12 studies published on the effects of creatine supplementation on brain creatine or brain phosphocreatine levels. 9 of these studies showed a difference, showed a statistically significant increase in brain creatine levels. Now, while this isn't dozens of studies, like with the muscle creatine literature, this is a consistent finding. We don't really have an explanation for why those 3 studies did not show a significant effect. It could have to do with certain characteristics of the research volunteers. For instance, their diet, their exercise, their age. But we do know that the literature at this moment points to the fact that the brain can be creatine loaded. We also believe at this point that the changes in muscle creatine are much larger than the changes in brain creatine. Although there's really only one study that has addressed this directly, and that's the paper from Solis and colleagues, where they measured simultaneous changes in muscle phosphocreatine and brain creatine. So in their investigation, muscle phosphocreatine increased across a range of plus 10% to about plus 28%. And in that same group of individuals, brain creatine increased from about 0% to about 4%. So the increase in brain creatine is absolutely lower, but we really don't know much about what explains the variability in terms of the response, or why the response is so much smaller in general. And this was seen in the original study by Deschent and colleagues. If you go back to this original paper, now they used a much higher dose of supplementation. It was 20 grams per day for 4 weeks. But the values in this study are not unlike the values in the Solis and colleagues study. You see some individuals had a 3% increase in brain creatine, another individual had a 5%, and then 2 what appears to be high responders, 13 and 12% increases. But this is still less than what we see in skeletal muscle.
How's it going? How's drop off?
Also,
yeah,
a reminder that in terms of variability throughout the brain, we do know that there are differences in creatine values in gray matter, white matter, cerebellum, thalamus, and differences in the response as well. So there are a lot of reasons to be cautious with creatine supplementation in brain research, because brain creatine metabolism is simply not the same as muscle creatine metabolism. Now, if you do ingest creatine and your brain creatine levels increase, what would we expect to happen? Again, we have a small number of studies, far less than what we know about exercise performance in terms of the skeletal muscle response to creatine supplements. But we, again, have a very, very consistent finding. To date, there are 16 studies that have examined the effects of creatine supplementation on cognitive processing. 13 of them reported an increase in cognitive performance, and 3 of them showed no effect. This is a rather consistent finding. Now, if you, for an in-depth dive to the comparisons of these studies, you can check any of our reviews, including the recent reviews published in Nutrients, or the excellent systematic review published in Experimental Gerontology in 2018. But for today, I want to make a few points to summarize this body of literature. And one point is that these articles are very difficult to compare. They are extremely different. For instance, in some of these investigations, the research volunteers were older adults, frail older adults. In other investigations, the population being studied was young, healthy adults. So I think what we've learned throughout today's presentations is that there can be differences in body creatine levels between older and younger adults. So it makes these studies very difficult to compare. In some cases, older adults were studied, in some cases, younger adults, in some cases, vegetarians were studied, while in other cases, omnivores or meat eaters were studied. So there can be large differences in the populations between these 16 studies, making for difficult comparisons. I think one of the most important points is that in some of these investigations, the subjects were well rested, while in other cases, they were very, very stressed, and not even stressed in the same way. In 2 investigations, the research volunteers were stressed by sleep deprivation and exercise, intense exercise. In those studies, the disruptions to cognitive processing caused by the sleep deprivation and stressful exercise were attenuated with creatine supplementation. In other investigations, the subjects are stressed mentally, where they have to do a demanding cognitive task for a great length of time. And again, in those cases, it appears that creatine supplementation rescues these individuals from a cognitive decline under these stressed conditions. Still, in other studies, they're young, healthy individuals who are well rested. They're not cognitively stressed. They're not physically stressed. In addition to the population differences and the research design, in this body of work, there are different outcome measures in terms of how cognitive processing is measured. There are different dosing strategies, different supplement durations as well. And I think we've done a good job highlighting this in these recent review article summaries. What I will say is that it's incredibly impressive to me that in a small body of literature, 16 studies, with all of these differences in population, with all of these differences in design, that the results have been overwhelmingly in favor of creatine supplementation improving cognitive processing. So 13 out of 16 studies showed an effect with wildly different methods. I'm impressed that with all of this noise in the data, that there is still very obviously an effect of creatine supplements on cognitive processing. So I'm impressed, and I hope this body of literature continues to grow and improve in the coming years. I'd like to spend a few more moments talking about the complexity of studying creatine metabolism as it pertains to the brain. And as an example, I'd like to show you what generally happens when we supplement individuals with creatine and we measure their muscle creatine changes. Here's an excellent paper published in Amino Acids in 2012. I want you to focus on the left side of the graph inside the red box. These 2 graphs show us pre to post supplementation muscle phosphocreatine in the placebo supplemented group and in the creatine supplemented group. On the top panel, you see the individual responses. On the bottom panel, you see the average response. So on the top panel, if you look at the slopes of those lines, you can see that the creatine group, each individual has an increase in muscle phosphocreatine as a result of supplementation. It could be a small, a medium, or a large change, but everybody increases in muscle phosphocreatine. And on the placebo side of the graph, you really don't see that sort of trend. Obviously, when you look at the average change on the bottom graph, clearly muscle phosphocreatine increased in the creatine group, and there was really no change in the placebo group. But now look what happens when we examine brain creatine changes in response to supplementation. This is another excellent paper from Bruno Golano's team in Brazil. And this is brain creatine levels in response to supplementation in 3 different regions of the brain. So pre to post supplementation in 3 different regions of the brain. But what I've done is I've placed a red box over the figure legend so you can't tell which group is the placebo group and which group is the creatine group. Unless you're very familiar with this paper, I challenge you to stare at those individual responses and tell me with absolute confidence that you can visually see which group is the creatine group and which group is the placebo group. If we, for instance, looked at the top right graph, we see some individuals increasing brain phosphocreatine and some individuals decreasing brain phosphocreatine, while still other individuals have no change. If we look at the bottom left graph, we see large increases in brain phosphocreatine and large decreases. So measuring brain phosphocreatine in response to supplementation is not nearly the same as measuring skeletal muscle creatine in response to supplementation. There seems to be a lot of variability in the response to the supplement, and we're not really sure why just yet. One of the biggest influencing factors on muscle creatine is dietary creatine intake. We've known this for many years. Vegetarians have less creatine in their body. They have lower blood creatine, they have lower muscle creatine. We also know that we can induce decreased body levels of creatine by simply transitioning an individual to a lacto, ovo, or a meat-free vegetarian diet. So I can reduce your muscle creatine levels in less than a month by transitioning you to a meat-free diet. We've known this for a long time. And here's a typical graph. We could use a number of different articles to make this point, but we do believe that vegetarians have less muscle creatine and as a consequence have a larger response to creatine supplements. So again, I'll point to this wonderful paper from Solis and colleagues published in 2017, which shows on the left side that omnivores do have a significant increase in muscle phosphocreatine when they supplement, but that vegetarians are starting at a lower level and appear to have a larger response to the supplement. So when we study skeletal muscle, everything makes sense in terms of dietary creatine and the response to increasing the amount of creatine in the diet or with supplementation. But let's look at the brain. Also from Bruno Gualano's lab. On the right, we have two graphs. The top graph is dietary creatine intake. Obviously, vegetarians have very little creatine intake from food sources, and meat eaters consume typically about 1 gram per day. So the top graph is an expected finding. But the bottom graph is really quite interesting. The bottom graph is brain creatine content. And on average, vegetarians and omnivores have the exact same amount of creatine in their brains, despite the fact that they have radically different dietary creatine intakes. Now, my guess would be that when dietary creatine intake is low, the brain increases synthesis of creatine so that brain function remains constant and brain health remains unaffected. But there's another important point here, and that has to do with the presence of creatine transporters that explains some of what we see here on these two graphs. The creatine transporter exists in brain cells, meaning that creatine can move from one cell to another. However, the creatine transporter appears to either not exist or be in very, very low levels on the astrocytic feet, meaning that it's going to be very difficult to transport creatine through the blood-brain barrier, which probably explains why the brain has the ability to manufacture or to synthesize its own creatine. So while skeletal muscle is designed to take up exogenous creatine, the brain appears to be set up to maybe resist brain creatine, but possibly that's only under certain circumstances, which we'll talk about now. Now, some investigators have said, if it's so difficult to study brain creatine in humans, why don't you just study brain creatine metabolism using an animal model? And I have to remind people that animal models have a lot to offer us, but in many cases, they can't be generalized to the human population. Now, here's a few examples of how complicated it is to study creatine across different species. I'm going to use animations here instead of actual pictures of laboratory animals. When we supplement humans with creatine, we expect about a 20% increase in skeletal muscle creatine. When we supplement horses with creatine, it has no effect. When we supplement rats with creatine, in many cases, it appears not to increase in the muscle, but does increase in the liver, which does not happen in humans. And the species that seems to respond the best to creatine supplements in terms of muscle creatine uptake are actually dogs. So hopping from species to species can create a very confusing body of literature. And that's just in terms of muscle creatine. If we look at brain creatine levels, the picture gets even more complicated. For instance, guinea pigs, mice, and rats respond radically differently to brain creatine uptake, showing a 54%, 32%, and 30% increase. Compare that to the human response, which is 10%, maybe, with more studies showing that this value is around 5%. So brain creatine supplementation research is difficult and is somewhat held back by the fact that there just aren't that many labs who have the capability to measure brain creatine levels in human beings. And jumping to animal models might not be the answer. So at this point in the lecture, some people get frustrated and they really want to ask me, does creatine get into the brain or not? And there are two answers here, really. Yes, 9 out of 12 studies show that this is true. When humans ingest adequate amounts of creatine, their brain creatine levels increase. But the transporter data suggests that this is very, very unlikely. I think this question has to be nuanced. I think we need to place things in their proper context. This figures in our 2019 review with Amir Dolan as the first author. And what it does is it proposes two different situations where creatine supplementation might be important to brain health. On the top, we have the example of preventive creatine supplementation. In this case, we would have brain creatine levels that could be insufficient, sufficient, or there could be enough creatine so that we could have a reserve. If a healthy individual takes creatine supplements, they may be able to increase their brain creatine levels slightly, creating a reserve such that when you stress this individual with intense exercise, sleep deprivation, hypoxia, some sort of acute temporary stress, that their brain creatine levels may only decrease into a zone that could still be considered sufficient or adequate. So there's one way creatine could benefit brain metabolism in healthy individuals. On the bottom, our proposal for therapeutic creatine supplementation features the same categories, insufficient levels of brain creatine, sufficient levels, or a reserve. And in these individuals who maybe have a cerebral creatine deficiency, depression, schizophrenia, maybe physical frailty and very old age, some sort of chronic stress that reduces brain creatine down to insufficient levels, these individuals could maybe get back up into the sufficient range with creatine supplementation, really opening the doors for the beneficial effects of creatine on brain health to be noteworthy for all different types of populations. Young, healthy, physically active individuals, those in the military, and also including a variety of different patient populations. One of these instances is concussion or mild traumatic brain injury. Several different nutrients are being thought of and being studied in terms of reducing the severity of a concussion or helping in the recovery from a traumatic brain injury like a concussion. Briefly, if you were to diagram the neurometabolic cascade of events that happens with a traumatic brain injury, as seen on this slide, you would find that there is tremendous overlap between the neurometabolic cascade of events that happens with a concussion and creatine metabolism, featured on this slide in red. So when you have a traumatic brain injury, a concussion, there's this large-scale membrane depolarization, there's reduced blood flow, there's increased intracellular calcium. There's basically a energy crisis that spreads across the brain and places extra demands on ATP to repolarize the membranes or to prevent this, the spreading of this energy depression type state. Creatine plugs in all over this pathway in terms of offering potential benefit in this instance of a brain injury. Now, how do we make sense of all of this? Is creatine monohydrate a potential nutrient that could reduce the severity of or enhance the recovery from a mild traumatic brain injury? Here's the evidence as of today. We've known for a long time that creatine is neuroprotective. That's number one. Number two, we know that brain creatine levels decrease with a mild traumatic brain injury. So right away, there's interest in creatine in terms of having protective effects on the brain. We do know that creatine supplementation is effective in reducing damage resulting from a traumatic brain injury. But we mostly only know this from animal models. And I've already spent a great deal of time reminding people that animal research and creatine research are very, very complicated to put together. But we have the data. Traumatic and hypoxic models in animals show remarkably reduced tissue damage when the animals are creatine supplemented. There's one fantastic study published by Turner and colleagues in 2015. This is a hypoxic model in humans. Again, this is a way to induce temporarily cognitive processing deficits. And in this instance, those individuals who have cognitive processing deficits from the hypoxic air, they're rescued by creatine supplementation. Those deficits are attenuated. And there are also these two investigations which have been already discussed at this meeting. These are open label clinical studies in a hospital setting with traumatic brain injury in children. These were very sick children, but the creatine appeared to remarkably reduce the symptoms of their brain injuries. Now, if you add all of this to what I've described today in terms of brain creatine and cognitive processing in human studies, there's a very, very strong circumstantial case here to say that we should be considering creatine monohydrate as a therapeutic for mild traumatic brain injury. I think it's worth taking a bit of a closer look here because the evidence is not as circumstantial as some people think, and it's probably not as convincing as other people think. So let's take this slide and let's look a little bit more closely. Again, I'll point you to some of the reviews that have discussed this at length because we only have a few more minutes today. But why would I consider creatine an important nutrient for concussion? Look at the list of things that happen in the brain as a result of a brain injury. Altered ATP demand, reduced brain creatine, a cellular energy crisis, membrane disruption, calcium influx, nerve damage, mitochondrial dysfunction, oxidative stress, and inflammation. These are all things that we know creatine can impact positively. Additionally, there's a research team that has access to retired football players. And in National Football League retired football players who are symptomatic and who have had repeated head impacts in their career, much later in life, they have reduced brain creatine levels. This indicates that the effects of something like a concussion can actually cause later life derangements in brain energy metabolism, which again, I'll say could be rescued by additional dietary creatine. The animal data are animal data. They're not human data, but they are very convincing. the reduced brain damage following traumatic brain injury in mice. Tissue damage is 36% reduced, 50% less in the rats. I'll remind you that while humans have an increase in brain creatine of about 5 to 10% with supplementation, with animals, it's more like 30 to 50%. So it's difficult to generalize these data from animals to humans, but it's very optimistic to me. In terms of the human data, we have improved cognition, improved communication, improved self-care, personality, behavior, reductions in headaches, dizziness, fatigue in these children with traumatic brain injury in a hospital setting. Essentially, all of the things we know that are problematic post-concussion appear to be greatly reduced and improved with creatine supplements. I think at this point in time, the best way to look at this is to say, we have documented muscular performance benefits. It's an inexpensive supplement. It's widely available. It has an excellent safety profile. Is encouraging creatine supplementation to reduce damage from or enhance recovery from mild traumatic brain injury premature? I actually think that encouraging supplementation for those populations at high risk for a brain injury might actually be considered prudent. So just to finish up with a few summary slides here, when we think about creatine supplementation and metabolism, I'd like everyone to remember that this is really a pleiotropic nutrient. It has important actions in several different tissues. It's a lot more than just the creatine kinase reaction. I also like people to understand that we've been studying creatine, the nutrient, for over 200 years, the supplement for 100 years, and the brain supplement for 20 years. There are hundreds of excellent investigations supporting the safety and the efficacy of creatine supplements. This is certainly not new. Creatine monohydrate clearly improves the performance of brief, intense exercise, perhaps mostly by increasing muscle phosphocreatine and glycogen resynthesis. Creatine supplements may improve the performance of some longer duration exercises, maybe out for a couple of minutes. It likely assists in adaptation to exercise, something that Darren Kandau did a great job highlighting in his presentation. And in terms of the brain, I think it's very likely that creatine monohydrate supplements improve cognitive processing and brain energetics, but we're really unsure if this improves sport performance. That's a gap in the literature. Everything I'm talking about are laboratory outcome measures, and no one's really studying the stressed athlete in a real sporting situation. There's very few studies in that direction. I think the mild traumatic brain injury are promising, but the data are certainly lacking. And then I'll finish with, as a summary, a few directions that I'd like to see this body of research take. In terms of creatine supplementation and brain content, we really don't have the dose response studies that are common in other areas of research. We need dose response studies. We need more studies on regional differences in brain response to creatine. And what we really need are labs that can measure simultaneously both muscle and brain creatine levels, and also total creatine levels and phosphocreatine levels, because typically with the MRI technique, a lab is usually only equipped to do one or the other. But both total creatine and phosphocreatine are valuable pieces of information to have. In terms of the supplementation and brain performance studies, we really don't have many studies that measured cognitive processing and measured changes in brain creatine. It's almost as if they're two separate bodies of literature, the cognitive studies and the brain metabolism studies. We need labs that can do both. I really think the connection between improved brain performance and sports performance is something that's lacking, and that there's a lot of teams that could really do some excellent work in this area. And certainly we have a lot more to learn about concussion and mild traumatic brain injury. I think these clinical trials will be very hard to do, but I think we can probably learn a lot from some excellent case studies in this area. With that, I'll say thank you for your attention, and thank you to the organizers for such a kind invitation to speak at this conference. You can reach me at my email address at work@Messiah University. In terms of social media, you can find me on Twitter at Rawson PhD. Thank you.